Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 1st February 2021. As we are into the revision mode and we are revising the entire syllabus chapter after chapter, and the revision is in the form of test paper, and in this session we are solving test paper 2, and the chapter that concerns for us is plant growth and development. You may recall from your memory that from this chapter certain topics are deleted and only selected ones are there, that is the plant hormones. So this chapter has got a limited edition for your batch for the exam, for the annual exam. So primarily the aim of revising the chapter in the form of test paper is to make you familiarize with the topology of the questions and how to present your answers. And particularly the 5-mark question, how to present 5-mark question. Normal tendency of the students is to write pages after pages. It is not that way you should answer. You should be specific in your answers. You should write point wise and explain the point and substantiate your answer with a suitable diagram. So keeping that in the background, let's move on. So general instructions, all questions compulsory. Question number 1 to 3 carry 1 marks. Question number 4 to 6 carry 2 marks. Question number 7 and 8 3 marks, and of course long answer type question number 9. Now first we go to the questions followed by the answers. What is aleurone layer? Question 2. Name the growth regulator which was first isolated from the corn kernel and coconut milk. Question 3. What is the full form of IAA? Next, question 4. Both growth and differentiation in higher plants are open. I will repeat, both growth and differentiation in higher plants are open. Comment upon that. Question 5, important one. What is bolting? Which hormone is responsible for it? Question 6. Why is the term short plants a misnomer? Question 7. Differentiate between photoperiodism and vernalization. Expected question, vernalization from this chapter. Discuss the statement, the growth is measurable. Its quantification can be done. Question 9. What do you understand by the term development? In that 2. Explain the sequence of development process in a plant cell. Now take a paper, start writing the answers. You may take 30 to 40 minutes to answer these 9 questions, and wherever possible substantiate your answer with a diagram, labeled diagram, and come to this. Now compare your answers with the answers that I am providing to you. Question 1. Answer, it is special aleurone layer is a special tissue layer which surrounds the endosperm in maize grain. Question 2. Zeatin or cytokinin was first isolated from corn kernel and coconut milk. So this is the first hormone that is isolated. And the expansion, of course, IAA, I for indole, A for acetic, another A for acid, indole acetic acid. Question number 4. Growth and differentiation in plants are open, as all the cells as well as the tissues arising from the same meristem may possess different structures at maturity. The maturity is determined by the location of the cells or tissues. It may be at shoot apex, root apex, and cambium. Question number 5. Enormous elongation of internodes. Remember, the space between the 2 nodes is internode, resulting in increase in stem height. Gibberellins cause the plants to bolt and flower. So what is bolting? Enormous elongation of internodes. That's bolting. Question 6. These plants require a relatively short daylight period, usually 8 to 10 hours, and a continuous dark period about 14 to 16 hours for flowering. In short day plants, dark period is critical and must be continuous. They are known as long night plants. And the term short day plant is a misnomer with long nights. So basically we should call them as long night plants. Look at that. The dark period, continuous, it should be 14 to 16 hours. There only flowering will take place. So it is more appropriate to call them as long night plants. Right? Question 7. Photoperiodism. As the name says, photo means light, periodism is duration. Vernalization. Photoperiodism is the flowering response of the plants to the duration of light and dark period in the diurnal cycle. Vernalization prepares the plants for perceiving stimulus for flower induction by chilling treatment, cold treatment. Photoperiodism is in this stimulus perceived by green leaves only. Here vernalization stimulus is perceived by young embryos, meristems and even leaves. Under photoperiodism, in this flow region is produced under photoinductive conditions. Vernalan is produced by chilling treatment. Vernalan. And under photoperiodism, photoperiodic induction cannot be reversed by exposing non-inductive conditions, whereas in vernalization it can be reversed when maintained at higher temperature. Alter the temperature, change the temperature, reversibilities. GA3 has the capability to replace the requirements of photoinductive conditions in long day plants only. And here it is the question of GA3 can replace cold treatment to induce vernalization. So what is important is cold treatment in under vernalization. Question 8. The growth is basically a consequence of increase in the amount of protoplasm. Since we cannot measure growth directly, it is measured by some quantity that is more or less proportional to it. So the growth is measured by variety of parameters like increase in fresh weight, dry weight, length, area, volume and number of cells, cell numbers. One single maize root apical meristem may give rise to more than 17,500 new cells per hour. What a great thing. 17,500 new cells per hour. So you can imagine per minute, 17,500 by 60 minutes. So almost or 6 3 yeah. The cells in watermelon can increase its size up to, look at that, 3,50,000 times. Therefore, growth may be expressed as increase in cell number or as increase in size of the cell. The growth of a pollen tube is measured in terms of length. An increase in surface area measures growth in dorsoventral leaf or dicot leaf. Question number 9. It's a term that includes all changes that an organism goes through during its life cycle, from germination of the seeds to senescence. Senescence is death. Diagrammatic representation of the sequence of processes in development of a cell in a higher plant is like this. So basically you have a cell division, cells divide, and then the cells divide, meristematic cell and plasmatic growth will be there. As a result you get a bunch of cells here, and the bunch of cells now start differentiating. Once they undergo differentiation, then there will be change in the shape and size, and that will be followed by expansion or elongation, and after that it is maturation. And once the cells get matured after undergoing differentiation, it enters into a stage called senescence, that is old age and death. So that's old age, senescence leads to death. So this is how the life cycle, sequence of development process in a plant cell take place. So basically it is the different stages that are involved in that. Basically it is the birth, then cell division, after cell division, formation of mass of cells, and the cells which are formed in large numbers to perform varieties of actions, functions, undergo differentiation. And once they undergo differentiation, they are no more similar appearing. Size will change, shape will change, cytoplasm content will change, location will change, and then after having undergone differentiation, cells will again start growing, and that leads to maturity. And a mature cell will survive for some time, perform its functions, and then enter into the old age, senescence, and the senescence leads to death. So this is how the cell cycle goes on. So in our next session, we will answer one more question.